And now we're going to go back to basic science again, and I'm delighted to welcome Dr. Nuria Peñuelas from here in Spain, who is going to address the question of neuromelanin and this age-old conundrum as to whether this is a friend or foe. Nuria.
Okay. Good morning, everybody. Thank you very much for giving me the opportunity to give this talk. It is a great honor for me to be able to present our work in front of this great audience. So today I will be presenting a new mouse model of Parkinson's disease that we have developed in our lab that reproduces the human brain-wide pigmentation and shows Parkinson-like pathology and transcriptomic alterations. I have nothing to disclose. So as you may know, in Parkinson's disease, there is a selective neuronal loss of neuromelanin-containing neurons, mainly loss in the substantia nigra and loss in the locus coeruleus. Neuromelanin granules accumulate with age in these specific cell types, and it has been hypothesized that might be contributing to their specific vulnerability to the disease. However, the exact role of neuromelanin accumulation in PD pathogenesis has remained just speculative because the most commonly used animal models in experimental research, like rats and mice, do not accumulate neuromelanin as humans do. So this is the reason why our lab, some years ago, developed the first in vivo model of neuromelanin accumulation by overexpressing the enzyme tyrosinase, which is a melanogenic enzyme, into the rat or mouse substantia nigra. With this study, we were able to induce human-like neuromelanin, and this neuromelanin accumulation in the substantia nigra of rodents induced neurodegeneration and motor deficits in these animals. However, neuromelanin is not restricted to the substantia nigra, but it is present at varying degrees in all catecholaminergic groups in the human brain, including the locus coeruleus and the dorsal vagal complex, which are also affected in Parkinson's disease. So our next step was to develop this new transgenic mouse model that overexpresses tyrosinase under the TH promoter, so that all catecholaminergic neurons would express tyrosinase and would thus accumulate neuromelanin. So indeed, when we process the brains of these animals, as you can see here, unlike wild-type mice, transgenic mice already showed macroscopic pigmentation in the substantia nigra, ventral tegmental area, locus coeruleus, and also in the dorsal vagal complex located in the medulla. And this neuromelanin was similar to the neuromelanin that we can find in the human brain in terms of being positive for or detectable by MRI, specific for neuromelanin, and also staining with Fontana-Mason stain, which is a specific stain for melanin. We also detected varying degrees in all catecholaminergic groups, thus reproducing the human brain-wide pigmentation. And we then quantified the pattern of neuromelanin accumulation through age. We defined different age groups, young, adult, and old mice, and we quantified the optical densitometry of the neuromelanin granules. And here we detected that neuromelanin accumulates in an age-dependent manner, and it follows a caudal rostral pattern, meaning that young animals showed higher, earlier levels of neuromelanin accumulation in the medulla and in the pons compared to the midbrain groups, while in the old animals, midbrain groups reach similar levels to the ones in more caudal groups. We then performed an histological characterization of the main three pigmented areas. We counted the TH-positive cells in the ventral tegmental area and in the substantia nigra, and detected a decrease in adult and old mice in both areas. However, this was due to a TH downregulation, since when we consider both the TH-positive and the TH-negative cells, which were still visible because of the accumulation of the pigment, we did not find any differences in the total number of dopaminergic cells, meaning that there is no neurodegeneration in these areas. However, we did confirm the loss of the dopaminergic phenotype by detecting decreased levels of dopamine release in the striatum of transgenic mice compared to wild types by performing in vivo microdialysis in collaboration with Analia Bortolotti's lab. Regarding the locus, we did find an earlier and greater loss of Th-positive cells compared to the substantia nigra and ventral tegmental area. And this was indeed neurodegeneration, since there was also a decrease in the total number of noradrenergic cells. And this great neurodegeneration was confirmed by the decreased levels also of total noradrenergic neurotransmitters in the locus and also in the prefrontal cortex, which is one of the main projection areas. We also characterized the dorsal vagal complex, which is this complex that consists in the dorsal nucleus of the vagus, which is cholinergic, and also the A2 catecholaminaergic group, which is the one that accumulates neuromelanin in this mouse model. And here we did not see any differences in the total number of Th-positive cells. However, when we focused on the pigmented neurons, which were around 10%, we did see a decrease with age, meaning that these neurons are also affected in this mouse model. Interestingly, we found typical PD hallmarks, including cytoplasmic inclusions, Lewy body-like inclusions, which were defined by P62 reactivity, and more or less 50% of them were also positive for synuclein. And we found this in all the main pigmented areas and exclusively in pigmented neurons. We also found extraneuronal neuromelanin granules and increased neuroinflammatory markers, including increased number of astrocytes, increased number of reactive or amyloid microglia, and increases in CD68 positive cells. We submitted these animals to an extensive behavioral characterization, and we observed some of the motor and non-motor symptoms that appear in people with Parkinson's disease, like alterations in the olfactory function, alterations in the sleep patterns, in voluntary movements, in anxiety and depressive-like behaviors, in cognitive tasks, and also in peripheral functions. We then performed a transcriptomic analysis of the main pigmented areas by isolating them by laser capture microdissection and performing transcriptomic microarrays. And here we detected that the main upregulated biological pathways in the three pigmented areas were related to the immune system, while the main downregulated ones were related to the RNA processing, proteasomal function, and mitochondrial function. These gene expression profiles correlated with gene expression profiles from PD postmortem studies. Interestingly, the correlation in the substantia nigra was found at 20 months of age, while in the locus coeruleus was already found at 3 months of age, further indicating that the locus coeruleus in this mouse model show the dysfunction in the locus coeruleus precedes the dysfunction in the substantia nigra in this mouse model. We then focused on the GPNMB gene, which was upregulated in the three main pigmented areas. And we focused on this gene because it is specifically relevant for Parkinson's disease. Since it has been genetically linked with the disease, it has been shown to be upregulated in the substantia nigra of people with PD postmortem brains. And also, it has been shown to participate in melanin synthesis in the periphery. So we further wanted to explore the role of GPNMB in a neuromelanin-induced PD-like context, and we first validated the GPNMB overexpression in transgenic mice and also in PD postmortem brains compared to controls, and then we overexpressed GPNMB in a neuromelanin-producing mice. And here we found a mild protective effect of the neogrostriatal system compared to the massive neurodegeneration that we observed in the neuromelanin-producing mice. This mild protective effect correlated with an improvement also of the motor performance of the GPNMB overexpressing mice as assessed by the cylinder test. So the key takeaways of this talk are that by modeling brain-wide pigmentation in vivo, we observe neuronal neurodegeneration and or dysfunction depending on the catecholaminergic group. We also see typical PD hallmarks, including Lewy body-like inclusions, extraneuronal neuromelanin, and neuroinflammatory markers. We observed multisystemic behavioral alterations, including both motor, non-motor, and also peripheral symptoms. We see molecular signatures that correlate with PD postmortem studies. And we also observe a GPN MB overexpression, which seems to be mildly protective in a neuromelanin-induced PD-like context. So overall, this new mouse model, since it reproduces typical neuropathological features of PD and also motor and non-motor alterations of the disease, it is an excellent opportunity to investigate new pathophysiological mechanisms and also to test new potential therapeutic targets. And finally, I would like to thank all my group, and especially my supervisors, Miquel Vila and Ariadna Laguna, and all of you for your attention. Thank you very much.
Nuria, thank you for such an interesting and extremely clear talk. Hugely promising findings there. Do we have any questions from the floor? So we've got one gentleman in the yellow at the back, and then we'll take your question over here.
Hi, yeah, that was some brilliant work here.
Okay.
I was just wondering whether you thought that the overexpression of the protein, whether the effects you saw was specific to the melatonin, or have you tried overexpressing other proteins in the same way to see whether you get the same results?
You mean like expressing other melanogenic enzymes?
Or overexpressing any other protein in the same pattern?
Well, we have tried to express other melanogenic enzymes that are known to participate in the same melanogenic pathway, and we do not see the same neuromelanin accumulation. So like TRP1 and TRP2, which also participate in melanin synthesis, and we do not see the same accumulation.
Okay, thank you.
Thank you. And another question from here at the front.
Very nice work, thank you. You know, we spend a lot of time focusing on the substantia nigra and the catechol-aminergic nuclei when we think about models of Parkinson's disease, for good reasons. But it's always fascinated me. Dr. Stossel here from Vancouver is laughing when I said that, but I'm going to refer to Vancouver now because the McGears pointed out so long ago that the basal forebrain cholinergic neural nuclei degenerate as much as, if not more, early on even, than the aminergic neural nuclei. And it seems to me that they might form a very interesting control area of the brain to look at in your animals. And I wonder if you had done that, and if you're by somehow seeing neurodegeneration there too, or if they're unaffected in this situation.
Yeah, this is an interesting question. We also asked ourselves that, and we checked the nucleus basalis of Meiner, cholinergic cells, in this mouse model, and we do see neurodegeneration at old ages. However, we didn't put it here because it's too much information, but yes, it does degenerate, even though they don't accumulate neuromelanin in these areas.
Fascinating.
Absolutely. I'm afraid in the interest of time and as a courtesy to the next speaker, we won't be able to take any further questions.